Hello, everyone. Very good seeing you all at the final CUSP conference. It has indeed been um, a journey from the start of the cluster until the end, and a lot of very interesting results, more questions than answers. I have a pleasure to present our project that also had uh, a focused approach in mind. We wanted to improve our exposure assessment, but also to understand the relationship of micronanoplastics with allergic diseases, and eventually to evaluate the role of micronanoplastics as vectors for contaminants. Allergies and asthma are chronic diseases, and there is no cure for allergies and asthma. There is a vulnerable population of children that shows higher prevalence of allergies, especially food allergies. That's why our project also had a focus on this vulnerable population, especially in the part of the clinical study we have conducted. So in a way, we followed the work of Aurora. When it comes to the whole project approach and our objectives, of course, we needed some new tools. At the beginning of the project, we did not have all the materials and the particles in sizes we needed. So we had to work towards getting the particles for the in vitro and in vivo experiments and the hazard assessment. The models, how to evaluate this specific hazard related to allergies and asthma, also had to be developed. So we have been working towards the models as well to evaluate the hazard and also to include the sizes, the micro and the nanoplastics as well. The routes we have chosen, the routes of exposure to micronanoplastics were also the routes of exposure to allergens, the inhalation and the ingestion. The only route we did not take into account is intradermal route, because at that time it was assumed that the route of exposure to the skin is not very relevant for micronanoplastics. Maybe it's something to look forward to and to also include because of a lot of particles that are present in the cosmetics. So this inhalation and ingestion routes of co-exposure of allergens and the particles were also tackled in our project, because we looked at certain matrices, such as food matrices, which are the rich source of potent allergens, such as seafood. And finally, we also wanted to evaluate the cargo effect. Can the micronanoplastics carry some harmful substances, especially those that are potent, and can mitigate and modulate the allergic responses? Of course, it also included the binding of the allergens, the proteins that are causing allergies as well. So we were going towards a building and health outcome assessment framework. And the key features, I think, have already presented of our health outcome assessment framework, where it has a focus on hazards relevant to allergy outcomes, and also it was a targeted assessment. So the hazards, the models we incorporated were those relevant for allergies and asthma. And of course, we wanted to integrate different components into this risk assessment data, such as the hazard characterization, which also includes, in a part, the particles itself, but also the allergens that are relevant for the IMPTOX in a way, how we thought about it at the beginning. We wanted to look into the dose response assessment, the exposure assessment, and finally to look into some health effects. So there are four key components of our framework: the particles characterization, the contaminants effect assessment, the exposure assessment, and the health effect assessment. The characterization of the particles also included the production of the particles. As I said, we were a very focused project. Instead of testing all the particles, which we realized we could not, we focused on the polypropylene and PET, thinking that those could be the particles, the polymer types that maybe children were the most exposed to. And we wanted to provide a well-characterized material to also improve some analytical methods to investigate the exposure and to assess different hazard characteristics, such as maybe the hydrophobicity of that material, and also to produce the material for in vitro and in vivo testing. It was very demanding, especially for the nano-sized materials. And our approach, thanks to the team from University of Vienna, it was actually very smart. We have designed materials, especially nanoplastic materials, that were from the beginning designed to be applied in different biological tests, in vitro or in vivo, meaning that the final product had to be biosafe in a way that there is no LPS. It was also provided in a medium such as glycerol, which is compatible with most of the cells instead of some other toxic organic materials. So this is a very comprehensive approach that will soon be published in Environmental Science Nano. Also, the University of Vienna team looked into the exposure to the occurrences of micronanoplastics in sea spray and lakeside aerosols. And this innovative sampling method was very actually good approach because it collected the aerosols into two fractions, the one we can inhale and the other one we cannot. And in these chambers, the collected particles were subject to pyrolysis GCMS. And in the samples that were collected near the lake in Austria, we could not detect any micronanoplastics. So it's a good news. We can say that the exposure is actually very low, but it does not mean that it will be like this in urban areas for which we don't have the data. We have also looked into the microplastics in different species and different regions of the allergenic foods, such as seafood. So we collected samples from Europe, 2 locations, but from the markets, also from South Korea. We have developed a sample preparation protocol that can allow us to quantify using micro FTIR particles. And we have achieved the kind of exposure assessment to the micronanoplastics from seafood. Because in our project, we also deal with the co-exposure. We needed to quantify the allergens, and we looked into the major allergens, such as the tropomyosin. We had to develop separate tests for the quantification of all of these. And what we have observed is that the foods with the highest occurrences of micronanoplastics has the lowest occurrences of the allergenic tropomyosins. So it is the shrimps with the lowest micronanoplastics that has the highest allergenic proteins, and in a way, this is a good news. We have also managed to develop a method to track the uptake of nanoparticles into basil. This is the model food that we have analyzed. We have observed uptake from root into stems, and all these data helped us model exposure to food ingested through shellfish microplastics. In the worst case scenario, we see that the number of particles is not that high, but we are indeed exposed. So these are the kind of conclusions from our exposure studies. Probably the most relevant conclusion is to say that the conclusions regarding the risk is impaired by the lack of health-based guidance values. So we cannot say that this number of microplastics is high or low because we honestly do not know what it means for the overall health. When it comes to the effect of the contaminants, we have analyzed the microorganisms, of course, allergens, toxins, and metals in different setups. And the hazard was assessed in vitro at the molecular level and also in the first line of defense, particularly immune cells, primary, and also some of the available cell lines, also the lung cells, and the gut, CACO2, especially HEP G2 cell line. You can see that in some of the in vitro tests that we have tested and used, we have observed the effects that could be potentiating the immune responses, the allergic, that could be harmful in a way. But I have to say, there is a dose response that we need to follow, and we say that in high doses, of course, we see more effects. In low doses, we don't see the effects that could be marked as harmful. So when we look in particular the allergic responses to allergen and the micro nanoplastics, these antibody binding to the proteins, the allergens present in the corona, is almost the same as an allergen, but we can say that there is a variety of exposed epitopes. But the effector stage, how these proteins, allergens, when present in corona, initiate degranulation of basophils, appeared a bit different. In all the individually tested patients, ex vivo, with cells isolated for allergic patients, we can say that the immobilized allergen on a particle is recognized by the effector cell, and it degranulates the basophils with a higher magnitude. So like this immobilization of allergen helps its cross-linking by the Ig antibodies and the effector's response. Of course, this is a bad news because it means more allergens taken up with the particles, a stronger response from the effector cells, and worsen clinical picture. We have also analyzed the response of T cells, and in these cases, we see no particular responses between allergic and healthy, but we can see the response of cells to the particles coated with the allergen. So finally, there is hazard when we look at the in vitro ex vivo tests we have analyzed, and the particles, if they serve as a carrier of allergen, could augment the allergic response. The magnitude of the response depends, of course, on the number of particles that the patient, the person is co-exposed with. Moving into the in vivo models, we also wanted to tackle ingestion, inhalation, but also we wanted to look into the systemic response, the immunization with the allergen protein and the particle, and a bit of tracking in vivo, because when you sacrifice so many animals, you really want to have as many parameters as possible. And we analyzed the respiratory allergens such as ragweed, the food allergens, tropomyosin, and chicken and white. And for this, we had to develop different models for systemic immunization, ingestion, inhalation, and also for tracking, acute and chronic. And the key findings are, again, the magnitude of the in vivo responses also depends on the dosage. And I could say that the doses applied are high. And in these cases, we see a systemic effect on the allergen-specific responses. We see the adjuvant effect. Then we see more severe asthma in case of inhaled PET, and also more intense lung inflammation in cases of inhaled polypropylene. We have also tracked the particles and developed a quantitative method to observe in acute and the chronically. In chronic exposure, we divided the dosage into 5, and we followed it in time. So we can see that the nanoparticles are quickly taken up from valve into lungs, but then there is also a quick elimination from the lungs. Our method is not very sensitive, so we cannot say much about spleen and liver and blood. But we have seen that there is this impact of plastics, chronic exposure on organ growth, on liver, on chronically exposed animals. You see actually delayed growth in comparison to the healthy, and this is the same both for allergic and chronic healthy exposed. So finally, into the health effect assessment, we also wanted to include our biomonitoring clinical study, where we combined the exposure to plastics information that we collected from children via questionnaire that collects information on the plastic exposure and some habits. We also did some biomonitoring on a randomly selected population of around 60 patients. And we wanted to screen, of course, the allergy association to the plastic exposure and also the occurrences of micro and nanoplastics. So for this study, we have collected around 110 samples and recruited more than 1,000 children in Croatia from 3 different regions, from the capital, the rural area, and the seaside area of Croatia. And we recruited both allergic and healthy subjects. It is interesting to see that even though Croatia is a small country, there are 3 different populations, 3 different regions, and they show differences in sensitization patterns. We see more respiratory allergies in seaside population, more food allergy in the capital. What we have seen through the plastic exposure questionnaire, if we look and cluster all the children into 3 populations, 3 clusters, the high exposure, medium, and the low. Around 60% of all children is in the medium cluster. There is around 30% of children with high exposure cluster. And there is an association of respiratory allergies in the clusters of children that are in a high and medium exposed clusters to inhaled allergens, and to food allergies in a cluster of children that are in a group of highly exposed, looking into the plastic questionnaire. We have also done the correlation of the fecal microplastics with the plastic exposure. And here we also see, even though these groups are small, it's only 56 analyzed samples. Here we can see that the groups differ and that there is the higher occurrences of the fecal microplastics in samples from the capital, where we also see highest food allergy rate. We can also say that even though the sample size is relatively small, it's only 56, it's still large in comparison to other studies conducted in stool. We already see what are the main causes of micro nanoplastics, what kind of polymers we can find most frequently in the stool samples of children, and these are the polypropylene and the polyethylene. In this way, we believe that we can guide parents if they want to decrease the exposure to micro nanoplastics, in particular in the allergic children, what kind of packages, food packages they should avoid, because the types of polymers we have found suggest that the main source of contamination comes from food packaging. So overall, our framework risk is building up. We have some key findings that are related to exposure, but still not complete because we lack data on nanoplastics and on more samples. We also see some hazards related that are targeted and related to allergies and asthma, but we still have a lot of bottlenecks and missing things, particularly related to methodology, the risk characterization, and the risk-benefit analysis to be able to talk about the risk and the health outcomes. So in a summary, we could say that we do not currently have all the information needed for completely risk assessment of any of the many different types, let alone environmentally relevant complex mixtures of micro nanoplastics. But we have improved our models, and we can say that by looking into the associations we have found in one population, we can model associations in others. So I would like to thank to all my partners in the consortium, 12 of them, and of course the CUSP colleagues, because we have exchanged a lot of information, material, and we really worked all together in this. Thank you.